we're just going to do some minor edits in the schedule, so we'll stay on time. So we're going to finish on time. Next speaker is my colleague, Dr. Andrew Malone. He joined WashU at the same time in 2014. He joined actually as at that time as a research or a faculty research fellow. A few months later, Dan told him that you need to do a clinical fellowship because the fellow left at that time. Andrew has been a great colleague for the last 10 years. He's leaving to Ireland in a couple of weeks. It's going to be a big loss for WashU. This probably his last talk as a WashU, but still after departure we're going to maintain very strong ties. And today we'll talk about the impact of APOL1, which still is a very hot topic. So excited about this talk. Thank you, Andrew.
Thank you very much, Tarek. Yeah, it's been a pleasure working in WashU over the last 10 years and learning from the great Dan Brennan and others. It's a really rich environment, both clinically and research, so hopefully they can continue to be a part of the family. So I've been asked to talk about APOL1 and its impact and relevance in kidney function. There is a talk that follows on APOL1, which I think will focus more on transplantation. I'm going to talk not about transplantation. I have no disclosures and no financially relevant issues with regards to this talk. So, firstly, APOL1 really hit the kidney scene about 13 years ago in 2010, when there was a number of big publications, in particular this one in Science in 2010, when studies were done to look at what genetic variants associated with certain flavors of kidney disease, in particular those related to African Americans. And this study was a genome-wide association analysis looking at end-stage kidney disease caused by non-diabetic etiologies in African Americans, and found that there was a number of variants associated with these renal clinical outcomes. And these two variants that I've shown here on the top left were highly associated with these clinical syndromes, and these were found in a gene called APOL1. There was actually two variants found very close to one another, two amino acid substitutions, and found to be in perfect disequilibrium, linkage disequilibrium, which means that they were always found together on the same haplotype, and we've now referred to those as G1 going forward. What this study then did was to regress out the G1 variants and reanalyze this from a genome-wide association perspective, and found another variant which was also very significantly associated with these pathologies, and was a deletion close to where the G1 mutations were, and we now refer to these as G2. Then to tidy things up, they regressed out G1, regressed out G2, and found no other variants across the genome associated with these two pathologies in African Americans. And so, looking at these variants, if you had two of these now called high-risk variants compared to none, you had an odds ratio risk of end-stage kidney disease from non-diabetic causes of 7. So what is APOL1? APOL1 is a serum factor, which is a trypanolytic factor found in humans, which confers resistance to a trypanosome parasite, Trypanosome brucei. The way it does that is APOL1 can insert itself into membranes, in particular in the trypanosome and the membrane of a lysosome. It acts as a channel. It swells the lysosome. It swells to the point where the lysosome bursts and kills the parasite. So TB fought back and decided to evolve into two subspecies of trypanosome, TB rodentia and TB gambiense. And these two parasites came up with the mechanisms to fight back against the APOL1 mechanism. And this is really well understood in the rodentia subvariant of TB. That's Trypanosome brucei. And it could secrete this protein called serum resistance-associated protein. It binds to APOL1 and stops it doing its function and confers resistance to being killed by the human, and so therefore could infect a human and cause African sleeping sickness. The exact mechanism of how TB gambiense does this is not really well known and still under study. So Looking at the variants that we found associated with end-stage kidney disease, it's interesting that these G1 and G2 mutations are in a region of the APOL1 protein called the SRA binding region. And so it turns out that in the G2 situation, if you have a G2 mutation in APOL1, SRA that the trypanosome makes can't bind to APOL1, and therefore APOL1 can then go and do its job, swell a lysosome, kill the trypanosome. And so humans fought back, and if you have these variants where you can therefore become resistant to African sleeping sickness, which is a huge pathology in the African continent. So in vitro studies have been done on this, and G2 human serum can kill trypanosomes in vitro. On the right-hand side here, you can see a graphic from a really good paper done in 2011 in JCI, looking at the kind of epidemiology of these variants in Africa. And you can see that in West Africa, the G1 in particular, but both G1 and G2 variants are really in high prevalence. They're prevalent in East Africa as well, but not to the same degree. And this roughly divides along the two subspecies of TB, Gambenzi in West Africa and Rodentia in East Africa. And as I mentioned, lots of in vitro studies done here on the bottom left. It just illustrates the fact that we know that if you have a G2 mutation, SRA can't bind to that protein. Well, what about APOL1 in relation to kidney disease, or kidney function rather? It's assumed and now thought to be true that APOL1 is not required for normal kidney function, and that's based on the fact that in higher order mammals, the APOL1 is not even present in the genome, not even as a pseudogene. For example, not present in chimpanzees and orangutans and macaques, and so they all have normal kidney function, very similar to the human. So assumed not required for normal kidney function. A really good review done by COP, etc. recently in Nature Reviews Nephrology is where I got this slide. It kind of really nicely highlights the structure of APOL1 and its relevance. At the top here you can see that these two mutations are found in a location that binds these serum-resistant factors that TB, Trypanosome brucei, secrete to try and defend themselves against human APOL1. On the bottom right, you can see whether you have a G1 mutation or G2 mutation, and whether that defends you against African sleeping sickness. So if you have a G2 mutation, you're pretty well, it's pretty protected against TB Rodentia infection, but not so much in Gambizi infection, and perhaps that's why there's extra pressure to evolve a higher prevalence of G1 and G2 in West Africa, where the Gambenzi subvariant is more prevalent. And then on the right-hand side of this slide, you can see its relevance towards the development of CKD. If you have any combination of two high-risk variants, you're at increased risk of CKD. And then on the left, you can see the prevalence of the haplotype. The G1 haplotype is much more common. About 22% of African Americans have this haplotype. G1, or G2 rather, is a lot less prevalent. That's haplotype. So in terms of how many African Americans have two high-risk alleles, this paper shows us that there's about 13% of African Americans have two high-risk alleles, but they do not all develop end-stage kidney disease. And so this leads to the assumption that there's a second hit hypothesis where another insult is required for you then to go on and develop end-stage kidney disease, perhaps interferon gamma driven in the setting of a chronic infection such as HIVAN, where we started to see this early in the HIV epidemic. So you need some other second hit to develop the disease, even if you have high-risk alleles. What about APOL1? Where is it in the kidney and what does it do? I'll try and be brief. Lots of studies have looked at this, immunohistochemistry and fish analysis for looking for mRNA transcripts in the kidney. Bottom line is it's found in most kidney cell types according to these studies, including podocytes. One interesting study on the very right-hand side of this slide suggested that if you have an FSGS lesion, the APOL1 expression is reduced, but that's only on the basis of IHC in a small number of patients. So looking at a lot of the data that I have a bunch of biopsies from recurrent FSGS patients in transplantation and controls, and looking at mRNA transcript at the single cell transcriptomic level, you can see here that most cell types express APOL1, but it's significantly higher in endothelial cells and podocytes, which is shown statistically at the bottom here. What about mechanism? Also, a huge area of interest in the lab and in the clinic. Again, this review highlights nicely some of the theories. There's a bunch of mechanisms, and I won't go into them all, from mitochondrial dysfunction, ion channels, amplifying the pro-inflammatory response, etc. But one maybe that we need to think about is APOL1 acting as an ion channel. Actually, APOL1 is pH-sensitive ion channel, and a lot of the really good early work done on this actually was done down the road in a lab by our SLU colleagues. But this paper shows nicely the relevance of the pH environment and how APOL1 works as an ion channel. Essentially, if you reduce the pH environment, so to an acidic environment, it increases the ability to kill a cell. So there's more potassium flux across an APOL1 protein or channel, causing that cell to die in an in vitro setting. But if you make that APOL1 a G1 APOL1, so put the variants into that protein, this change in pH, or this acidification, amplifies the ability to kill that cell. And the reverse is shown on the right when you cause a basic environment. These G1 and G2 variants amplify, it's a gain of function mutation in terms of its ability to act as an ion channel. So what about treatment? And I'm going to wrap it up in the next couple of slides. But this paper was published a couple of months ago. You've probably all read about it. It's a really nice paper published in New England Journal. It's really translational. They have a bunch of in vitro studies, some mouse studies, and human studies. So they look at the effect of this drug, which is an ion channel blocker called inaxaplin, and its ability to act on APOL1 and potentially treat patients with proteinuria due to APOL1 gene mutations, these two high-risk alleles. So on the top you can see if you put this protein APOL1 into cells in a dish, kidney cells in a dish, and you measure how much ion fluxes across that cell membrane, thallium used as a surrogate for potassium, you can see with increased doses of inaxaplin that ability to flux ion, potassium is suppressed, and this suppression effect is greater when you have a G2 or a G1 mutation in the APOL1 protein. And the top right graph just shows you that it's APOL1, because if you don't have the protein in there, the in vitro model doesn't work. On the bottom here, what they did was they created a mouse. They made a APOL1 G2 homozygous transgenic mouse. That is, they put the APOL1 gene into the mouse. It's not normally present in there. They had 2 copies of G2 APOL1 on either chromosome, so it's homozygous. And their model was basically to give the mouse a bunch of interferon gamma. This causes proteinuria in a mouse. So if you do this as a control and then add inaxaplin, it shows you that you can suppress proteinuria in this mouse, which is shown on the bottom right-hand side. And then finally, in a small clinical trial, they had 16 participants in an open phase 2 clinical trial where they took patients who had biopsy-proven FSGS, 2 APOL1 variants, proteinuria, and GFR that wasn't rock bottom. And they gave this drug over 13 weeks and showed that they could suppress proteinuria over 13 weeks, which was pretty impressive. A really nice set of studies in this paper, and shows a lot of promise for something that we're only beginning to understand from a clinical point of view, and has a big impact in African American patients in terms of burden of kidney disease. And so to summarize, G2 high-risk variants, or a combination of G1 or G2 in APOL1, increases your risks of end-stage kidney disease that's non-diabetic in etiology, up to an odds ratio of 7 when compared to risk alleles versus 0. These variants evolved as a conference resistance to African sleeping sickness. APOL1 is not required for normal kidney function. It's mostly found in podocytes and endothelial cells. It's probably mainly acts as a gain of function ion channel and is pH sensitive, and perhaps we now have an effective treatment for patients with these variants. And with that, thank you very much for listening, and I'll take any questions.
Time for a question.
Great talk. My question is about patient with FSGS, young African American, proteinuric, and nephrotic syndrome. So edema, everything, like the type that we think can recur. Is there any chance of recurrence in a transplant in a patient like this? I'm not talking about the non-edematous type.
So someone who's got high-risk alleles in APOL1,
yes,
presumed driving the...
yes. Is there any risk? Is there any concern about, for example, circulating APOL?
Good question. There is some data in the transplant world actually that show, like, a lot of people in the transplant setting have focused on donor and how donor APOL1 high risks infer risk post-transplant. But there's data coming out in a good JCI paper by a group in New York and Yale that showed that actually recipient APOL1 high risk infers, independent of what the donor is, infers higher risk of rejection post-transplant, suggesting that there's some sort of immune cell mechanism related to G1, G2 variants in APOL1 or a serum-related factor increasing risk. But that's rejection and graft failure. Recurrent FSGS, I don't know the answer there. And then in the setting of a transplant, if the native kidney disease is thought to be driven by two high-risk alleles, and then you might want to know what the donor is, or it's a non-living related kidney, that might mitigate some of your risk, but perhaps not to zero.
I'm just going to follow up on the point that you just mentioned. That means possibly if the recipient has two risk alleles, we're going to maybe give them a higher dose of thyme. Is that what the conclusion is of the study that you're mentioning?
Yeah, I don't know. I think it's just a signal. I think there was that JCI paper a couple of years ago. I think others are starting to talk about it. There might be other smaller another smaller study that suggests that signal. The exact mechanism of it, for example, is it T-cell driven, which is what thymo is all about. Nobody knows, and so I wouldn't jump on that and assume you just need more thymo. Like,
I mean, higher risk of rejection.
Well, higher risk of rejection, yeah, more thymo, but this is related to APOL1 risk variants and how that mechanism, it's not necessarily HLA related, or who knows. But bottom line is that I wouldn't jump to assuming you need to give more thymo based on that. But the mechanism, who knows?
Thank you.
Thanks.